Bipolar disorder, also known as BPD, is a psychiatric disorder defined by recurrent extreme mood swings. Some cases of BPD are induced by drugs or alcohol or due to other medical conditions. However, bipolar disorder can also be caused by genetic variations. The heritability of BPD is around 70%. BPD has around 21 SNPs that can affect an individual's risk of inheriting the disorder. For example, VAL66 shows an increased 1.7 risk with the allele combination AA compared to the standard allele combination of GG, which entails no risk. Genetic differences attributed to BPD are typically located in the brain and its cells, which makes sense due to BPD being a psychiatric disorder. Our group investigated the SNPs RS3847953, RS933399, RS775737, and RS6265. Of these four, we believe RS775737 and RS6265 to be the best predictors of BPD. But what do these numbers and letters mean? A SNP is a single nucleotide polymorphism, meaning that every SNP is a variation in the nucleotide sequence of DNA. So a C, cytosine nucleotide, in one person may be a T, thymine, in another person. The RS before each set on numbers for the SNP stand for reference SNP, and the number as the identification number. RS775737 is a SNP in the FKBP5 gene and is associated with an increased risk in BPD for a person with that SNP. FKBP5 influences glucocorticoid function. It does make sense for these two SNPs to affect the risk of BPD, especially the FKBP5, as its function is linked to many psychiatric disorders. According to dictionary.com, a glucocorticoid is any corticosteroid that is involved in the metabolism of carbohydrates, lipids, and proteins. If the glucocorticoid receptor is impaired, levels of hormones such as dexamethasone and cortisol will fluctuate with side effects that can impair mood and stress levels. RS6265 is the second SNP we researched, and this SNP is linked to the BDNF gene, which when affected by the RS6265 SNP has a linkage to BPD. BDNF stands for Brain-Derived Neurotrophic Factor and is a protein that is encoded by the BDNF gene. Polymorphisms in this gene lend to an increased susceptibility to BPD. VAL66 is the gene associated with RS6265 and is a polymorphism of BDNF. The AA and AG genotypes are associated with decreased brain motor function, introversion, and increased risk for depression and BPD. There are many benefits of being tested for BPD, but notably is testing for BDNF and its relationship to the VAL66 allele because the allele alters the efficiency of BDNF secretion, which leads to decreased gray matter growth, as BDNF controls cell and neuron survival and growth. Those diagnosed with bipolarity have smaller amounts of gray matter. Although our SNP chip test is in your best interest, there are some limitations to this test. These limitations include the issue of not having substantial existing data for certain ethnic groups to provide a reliable and accurate result for one's test. The ethnic groups which SNPpedia has data on include Han Chinese, American Caucasian, Russian, Japanese, and English Caucasian. If your ethnicity does not match ones in this list, our SNP chip test may not be as valuable or reliable in terms of determining your likeliness for developing bipolar disorder. The major warning and ethical consideration for one participating in our SNP chip test is the mental effects which taking a test with such hard determining factors as this one. The psychological effect which the results can bring onto a patient can be difficult to handle, as a positive result is usually a maybe when it comes to SNP chip testing. The idea of the possibility of a patient developing BPD can cause paranoia and anxiety, as they may question when the BPD will set in, how it will affect their lives, and various other considerations which one must take after receiving our SNP chip test.